Hello biology students, this is Dr. B and we're working on why is a body temperature of 107 deadly? We are on activity 1 of this part of the unit. How do polar and nonpolar interactions affect protein structure and properties? We did pages 1 through 5 in the first video, and now we are working on pages 6 and beyond. So how does molecular shape affect interactions between nonpolar and polar molecules? Let's find out. We're going to explore the simulation and think about shape affects interaction between nonpolar and polar molecules. So let's select a pair of molecules here. These are nonpolar molecules, and we can see by how far the spring gets pulled out, how difficult they are to pull apart. Let's look at another set of molecules here. These are still nonpolar, and we have to pull the spring out a little bit more to get them apart. Let's reset that and do that again. So pay attention, these Z shapes here are representing a spring that we have to pull out before they separate, and we can see we're pulling the spring quite a ways out before these large straight molecules get separated. Let's look at bent, straight and bent. So this molecule is straight. This molecule is bent. So the contact here is here, here, and here. So there's only really 3 atoms that are contacting this molecule here. Let's look at how difficult this is to pull apart. So notice how far that spring pulls out. Let's go back and compare it to large straight molecules. And then let's go to straight and circular. There's just, you can see a couple atoms interacting with the straight here. Let's pull that out using the star. Looked like it was pretty easy to pull out, and obviously you should be working on this on your own if you can. And here, this is similar to an active site. In the last page, in the last video, we were talking about active sites and the substrate and an enzyme. And this is somewhat similar to what you'd see in an active site here. This would represent the larger enzyme, and this would represent the substrate. And let's pull this out. It's a little bit more difficult to get out here. Even though these are nonpolar and the interactions are temporary and not very strong, I still have to work a little bit harder to get this curved molecule out of a round molecule, or get the round out of the curved. So now we need to take a snapshot of the model showing the molecule you think would form the strongest attraction. I personally think this one is the strongest attraction here. Let's take a snapshot of this one. And some things you could use to formulate your answer would be curved, round, number of atoms interacting, and you could say strength of attraction. How well is this round molecule going to stay stuck in this curved molecule? That's really the question we're looking at. And here's a word bank you can use to formulate your answer. All right, let's go to the next one. It says, take a snapshot of the model showing which molecule you think would form the weakest attraction. And students in my class had a spirited debate about whether it was the 2 small molecules like this, because they pull apart really easily, or if it was the large straight and bent molecules like this, or it was the large straight circular molecule like this. Before you formulate your answer, think about how many atoms here are interacting with how many atoms here. We can look at that too in the small molecules. There are 2 atoms here interacting. That gets pulled apart very quickly. So think about the number of atoms interacting. I think the vote in our class was straight and circular. You can make up your own mind. I will take a snapshot of this one and give you some hints about things you should think about. Number of atoms interacting, type of interaction. So is it polar or non-polar? And just look at that spring that you're pulling on. The star is connected to a little model of a spring. And how far is the spring pulling out before it separates? That's some evidence you can use to support your conclusion here. Predict how the potential energy of the system will change when the molecules are close together or when they're far apart. And remember, we've talked a lot about our water molecules here. Water molecules are very, really good. They're very polar and they're very good at sticking together. When you separate, the potential energy of the system increases because there's some electrostatic force working on these two molecules trying to pull them together. So there's some potential energy of the system for these guys to get together. There's pull here. When they are together, that potential energy dissipates because they're already together. The potential energy has been converted into kinetic energy as they move together. So that will help you formulate your answer there. So you want to use words like potential energy, distance, increase, decrease. That's a good word bank to use when you're discussing question 16. So now is a great time to return to the Jello mystery. If you remember correctly, we made good old regular Jello. Here's the control. And we put a quarter on top to show that this Jello is nice and solid. It takes on the shape of its container and doesn't kind of pour out of the container when you turn it over. When we made Jello with boiled pineapple, I boiled the pineapple here, put it in the Jello, and made Jello. The quarter sits on top here. You can see the quarter is sitting on top. If I tip the Jello over, it's still a solid. It doesn't run out of the jar. This is the boiled pineapple. We're trying to solve the mystery of the pineapple here that's fresh pineapple. This fresh pineapple that did something to the Jello that's unusual. The quarter goes right through, and the Jello is not a solid. If I tip this here, it's a liquid and it's going to pour right out of the jar. So there's something different about the fresh pineapple here that makes the Jello different. This Jello is very liquid. This Jello with the boiled pineapple is very solid. With the liquid pineapple, with the fresh pineapple, it's a liquid. So let's think about a scientific explanation to account for our observations in the pineapple Jello experiment. Let's justify our answer using ideas related to protein structure, function, and energy. So let's think about kinetic energy added to the boiled pineapple. And let's think about the proteins in the boiled pineapple, how they might be different than fresh pineapple. Because the fresh pineapple was not heated. The boiled pineapple had a lot of extra kinetic energy added to it that may have affected those protein structures. If there are enzymes in this pineapple, they were not affected by the heat like they are in the pineapple here, the boiled pineapple that was heated and makes the Jello solid. The fresh pineapple that wasn't heated doesn't have extra kinetic energy added to the protein structure. That did not allow the Jello to solidify. So there must be something in the fresh pineapple about the protein structures in the fresh pineapple that's different here in the fresh pineapple, which makes kind of liquid Jello, than the boiled pineapple then makes solid Jello. And that probably has something to do with the extra kinetic energy we added that affected the protein structure of the boiled pineapple and how that protein structure, that enzyme structure, affected the Jello. Let's go to the next page. So let's look at how temperature affects protein folding. I'm gonna just start this so we have time to look here. Think about how temperature affects interactions with the proteins. Then answer the questions that follow. So we're really looking at how temperature affects protein folding here. What types of interactions can we observe in the final structure of the protein at low temperature? So this is the protein at low temperature. Let's take a snapshot of this so we have a reference before we turn up the heat. So we can say low kinetic energy. We haven't added a lot of heat. So these molecules don't have extra kinetic energy. We can say there's polar interacting with polar as the water and the polar amino acids. So polar water and polar amino acids interacting. We know that the nonpolar is in the middle because they got pushed there by the polar that want to be on the outside and interact with that polar water. Those polar amino acids, they want to be on the outside so they can interact with the other polar amino acids. So our polar amino acids here are green in my model, in the model on the screen. So they kind of push the yellow, the non-polar amino acids, into the middle of the protein so the polar amino acids can be on the outside. And this creates a very distinct protein structure. Every protein has kind of the correct protein structure to do its job, and then it has other structures that can happen as it's exposed to other different conditions that are different from its normal conditions. So now let's turn up the heat on this protein. What's happening here? Does it can maintain the same structure as we add kinetic energy, as we add movement as heat? We know that heat is a measure of the kinetic energy. So as we turn up the temperature, we turn up the kinetic energy, because temperature is simply a measure of the kinetic energy in a particular system. As we turn up the kinetic energy, we also turn up the potential energy as we pull those polar molecules apart. Our next question asks us, what types of interactions can you observe in the final structure of the protein at medium temperature? All right, let's go to medium temperature here. Let's reset, go to medium temperature and start it off. If we look at our snapshot here, this protein is a little bit looser and it's got definitely a lot more movement in it. So let's take a snapshot of that here. And while we're waiting, we can say more kinetic energy. Atoms are further apart. Polar amino acids not so organized. They might not be interacting with as much with each other or with the water as they are in the lower temperature molecule, if you look at the snapshot of the protein at a lower temperature, because there's less kinetic energy. We can see this is pretty organized and everything's pretty tight, and this is pretty looser. Those connections aren't as close to each other. Let's turn up the heat here and see what interactions we get here. Let's reset this and start again. So we can see the potential energy is pretty high. After a while, it does go a little bit lower, but it's very much fluctuating. And we can see that it's even a little bit more loose than it was at medium temperature. And it's definitely a little bigger, and there are looser connections than there were at low temperatures. And this has a kind of a constantly changing shape, where this shape was pretty definite and pretty constant at a lower temperature, much more constant than it is now at this high temperature. So let's take a snapshot at high temperature, and we can say more kinetic energy. We can say more space between the molecule, or between the atoms. We can say polar are not as organized. It's just kind of a looser, and it's changing and wiggling around a lot more as that kinetic energy of the polar forces that normally organize the model. As we add kinetic energy, things kind of get looser and the protein isn't quite as organized. Let's do that over again so I can actually get a snapshot. It helps, if you want to get a snapshot, you need to stop it and then take a snapshot there. And there's your word bank. So how does temperature affect protein folding? We know proteins are normally folded in a very specific way, so that polars on the outside and nonpolars on the inside. That's polar and nonpolar amino acids. When we add kinetic energy, things start to shake up and we don't have quite the same organization. Some of those bonds between the polar amino acids and the polar amino acids in water are changed because there's more kinetic energy, and that kinetic energy becomes greater than the energy in the bonds here. Remember, there's potential energy when we separate polar molecules, especially when we separate, we can see that in water. We know that water, the dipoles of water have electrostatic forces pulling them together, and that's the same with any polar molecule. So there's more potential energy here than there is potential energy here when they get together. So the energy between the polar and nonpolar that's usually present to organize a molecule at lower temperatures, that is overcome or less important compared to all the kinetic energy you add when you heat it and things kind of fall apart. So you can say kinetic energy. You can use the word overcomes. You can say polar bonds. You can say structure, and you should probably say change. Question 22. Do you think a protein can effectively perform its function at high temperatures? Well, let's take this model of a protein. The green here represents polar, it's trying to be on the outside. Purple's trying to be on the inside, and it's creating a very unique active site here. In the middle here, we have a very specific shaped active site. Let's find our molecule, our substrate that fits in the active site. So here's our substrate. It's going to sit in the active site, and this enzyme is going to act on this substrate here. So enzymes on the outside, substrates in the active site, and this is a working enzyme. If we add heat, the enzyme can change its structure. The active site isn't there anymore, and there's no place for that substrate to go. So that gives us some information about how changes in temperature affect protein structure and protein function. So we can say protein function, we can say structure changes, and function changes. Those are some good word bank words with you can construct your answer with. Question 23 asks us, how do changes in temperature relate to changes in energy of the system and structure of the protein? Justify your answer using ideas from the simulation. Well, let's watch what happens. So we've got a protein, let's get a new one here. We've got a protein at low temperature moving pretty slowly. The potential energy is high in the beginning, but as it forms and those polar molecules kind of find each other and connect, the potential energy goes down and it forms a stable structure. When those polar amino acids get together with water and with each other, we get a nice stable structure. That's a very specific structure, and it's a very stable structure because the potential energy is low. Once we start adding heat, the potential energy goes up because we loosen things up. Those polar amino acids get further apart, and anytime we have polar molecules further apart, there's those electrostatic forces trying to pull them together and creating potential energy. So you can also use the simulation here to reset, start. You can change the temperature. Watch what happens to the potential energy as you change the temperature. There's a nice stable protein, low potential energy, stable. All the polar molecules are interacting the way they're supposed to. The potential energy is low because they're together. The electrostatic forces aren't there trying to pull them apart. They're already together. If we add kinetic energy, that adds a different kind of energy versus the electrostatic forces that are trying to pull our polar molecules together. And it just adds more energy to the system, so it's different. These forces are still there that are trying to pull the polar molecules together, but there's enough kinetic energy in the system that electrostatic force trying to pull those molecules together, that potential energy, is small compared to the kinetic energy. So we can say kinetic energy changes in total energy of the system. As kinetic energy is transferred to the protein system, the total energy of the system goes up. The potential energy is a smaller fraction of total energy. So it's not that the potential energy between the polar molecules goes down. It's just as more energy joins the system, this potential energy is a smaller portion or a smaller fraction of the total energy. And so there's just more energy in the system, and that's causing the molecules to move more. It's kinetic energy, and that constant movement, it's like shaking my bag of water molecules here. They're stuck together right now, but if I shake them, I break some of those interactions because I add energy to the system. I transfer some of my chemical energy from my muscles to kinetic energy of my bag of molecules here, and I add energy to this system. I transfer energy to that system from my muscles. I have chemical energy that I ate. I transfer it into a mechanical energy, and that's kinetic energy here. That energy of the system goes up when I shake it. And so there's more energy total, and so the total fraction of potential energy is smaller. So it gets it's less important in the entire equation, energy equation. So we can say total, we can think about the energy sum of the whole system. All right, so that is page 8. We'll work on pages 9 and 10 in a video number 3. So this is Dr. B signing off. We did pages 6, 7, and 8 of activity 1 in Y is a body temperature of 107 Fahrenheit deadly. If we add energy, kinetic energy, to our own proteins and our own body, this sort of thing can start to happen to the molecules on our body. Our enzymes get shaken apart and they don't work, and that is damaging to us. So keep thinking about that as we progress in this lab.